Good, good morning to everyone. So I'm Fabrizio, I'm from Brussels, and today I will present our recent investigation on stability activity trade-off in enzymes. And this talk is based on this couple of papers that we published one in 2021 and in a recent GCTC that come out, and some also work in progress. So enzymes are key proteins in countless biotechnological applications because they are used as biocatalysts in food industry, textile, pharmaceutical industry. And they are characterized by a very well-known stability activity trade-off. In a certain sense, they are like us. I mean, we are stable. We are sitting here all the days, but at certain times also we need to move, go outside, take drink some things, do a bit of activity. On the same, in an analogous way, enzyme, they have to be stable because they have to fold in a stable 3D conformation, but they have also to perform their function. So they have not to be too stable because they have undergo conformational change. I mean, the open and the catalytic pocket, and then the substrate enter, bind with the enzymes, and then the product come out of the catalytic pocket. And so they do not have to be too stable. That in enzyme that there are regions that are optimized for protein function, but not at all from protein stability is known from the 80s when experimentalists did mutagenesis on the active site of the enzyme, and they soon realized that the activity of the enzyme is completely destroyed, but instead the stability of the enzyme is increased. And so, in general, this trade-off between stability and activity is a limitation about the application of enzyme biotech processes, because in biotech process we want, to work outside physiological condition in any condition of temperature, pH, and while the enzyme has to be as stable as active. And so the aim of this investigation is to use the computational approach that we define to better understand this trade-off, and then use what we learn in this investigation to in the future to improve enzyme design methods. So now let me say a few words about our computational approach, and in which in the first paper we define the notion of stability, strength, and weaknesses. So the idea behind is that proteins adopt their lowest free energy conformation in physiological condition, and this is true for globally for the protein structure. But if we focus on a given element, even a secondary structural element or a given residues, these individual elements are not necessarily in their lowest free energy conformation. In other words, we can have some residues that are happy to live in that conformation and then contribute positively to the global folding free energy, while there are other residues that are not so happy to live in that conformation and does not contribute to the global folding free energy. So how we quantify this notion of stability, strength, and weakness? So we use the formalism of statistical potential. It's a very well-known formalisms that are derived from non-redundant data set of well-resolved protein structure. In our case, we use a simplified representation of the protein structure in which we only consider the main chain atoms and the average side chain center. And so basically we have our data set of protein structure, and then from the probability of observation of certain structure element association in this data set, we have some energetic values. For the sequence element, we consider always the amino acid type. For the structure element, we consider three structural descriptors. One is the inter-residues distance, the second is the solvent accessibility of the given residues, and the third one is the torsion angle domain. So basically, the probability of observing this structure sequence element association in a data set is written down in terms of number of occurrences of this element in the data set, and then properly normalized to get these energetic values. Just a simple example, we have phenylalanine and tryptophan separated by 3.9 Armstrongs, just by counting how many of this configuration there are in our data set, how many of couple phenylalanine tryptophan there are in the same data set, and how many of couple of residues are separated by 3.9 Armstrong, we have the energetic values of this configuration. But in defining the stability, strength, and weakness, we want a per site indication. So what we did basically is we partitioned the energetic contribution of an amino acid per interaction equally to each of the two residues that are interacting. Again, I have the couple between the phenylalanine, couple of residues interacting between phenylalanine and a tryptophan. So what I do, I take the energetic values, and let's suppose the energetic value is 2 kilocalories per mole. I split one on the phenylalanine and one on the tryptophan. And then I repeat these tricks for all the residues by considering, the interaction between the phenylalanine and all the other residues in our proteins. And every time I split the energetic contribution half on the phenylalanine and half on the other residues, and then I sum together, and I come out with this per site, what we call per site folding free energy. Again, there are 3 descriptors, so we have 3 types of per site folding free energy that then we sum together to arrive to the final index that we call per site folding free energy. And this is the stability index that we use. So if this index is smaller than 0 in our convention, we call this residue stability strength. That means that this residue is happy to live in that conformation and contribute to stabilize the global fold, while if delta GE is bigger than 0, that means that that residue is not so happy to live in that conformation, and then doesn't contribute positively to the global fold, to the stability of the global fold of the enzymes. We also develop a web server called Swotains, in which you can input your PDB, and then the web server do automatically the computation. So take the energetic values and then compute and do all the splitting and output the values. We have also some visualization tool in which you can visualize indeed your protein and look visual. The region that are weak, the region that are strong, and so on. And we color according a certain scale. So if the color is dark blue, means that residue is it's very stable, so contribute strongly to the stability. While if instead is dark red, means that it's very weak. That residue is very weak. The web server, you can find it at that link, and we also publish some detail about the method in bioinformatics. But today I would like to present how we apply this method to the enzymes, and to study a bit the stability pattern that there are in the enzyme structure. So, of course, we start by creating an enzyme data set. We start from taking all the enzyme with known structure and known catalytic residues from the catalytic site atlas, and then we filter for sec ID and for structure resolution, and we check also the correct oligomeric structure that was not annotated in catalytic site atlas. And finally, we end up with about 550 enzymes with different AC classes. And then we apply Swotain to this data set of proteins by computing the delta GE for all the residues of all the enzymes on this data set. And this is what we get. So here I plot the average of delta GE over all the residues in our data set as a function of the distance of the residues to the closest catalytic site. So what we can learn from this curve is that, okay, if delta, when the distance is equal to 0, the contribution, we are basically in the catalytic residues, and their contribution is very weak. Then there is a first shell of residues in which they are not weak. They are neither strong, but they are certainly not weak. Then there is a second shell of residue that indeed seems to be weaker. And then from a certain distance, then all the residues in average are strength. So there is a sort of oscillatory pattern, but here we have to consider that in this curve, we basically average on a lot of enzymes, so enzymes that have different oligomeric structure, different length, different active sites with different catalytic sites. There are some catalytic sites, there are just few residues, others there are a lot. And so here the effects are a bit smoothed out. But if we look singularly to the distribution of delta G as a function, again, of the distance of the residue with respect to the catalytic site, this oscillation is much more clear. And it's clear that we have some things that is weak. I mean, the catalytic site are weak, but this was already known from a while. Then there is a first shell of residues that appear to be a strength, like a sort of compensation, while the second shell of residues that is again as weak as the catalytic site, and then of course the other residues far from the active site are in general strength. Of course, there is, again, here there is a bit of variability between the different enzymes, because, I mean, statistical potential usually define up to a certain constant related to the length of the protein, so we can expect a sort of variability. And also in the x-axis, probably due to the fact that these enzymes have a different active site with different size, so we are still investigating how we can take into account all this variability between the different enzymes. We also did some further check. For example, we checked that this behavior is independent on the AC class because, or in other words, it's independent on the catalytic reaction that the enzyme catalyzes. And indeed, this, I mean, plotting the same graph that the G as against the distance from the closest catalytic site, we observe the same in all the EC classes. And also separately from current surface, because we were thinking maybe this is a side effect of the residues distribution, solvent accessibility residues distribution, because we know that surface and core residues contribute different to the stability, but I mean the curve appear similar and seems present in both regions. So this is as a sort of schematical picture, what we found, basically in the sticks are represented the catalytic residues and are represented in dark red. That means that these are very weak residues. Then there is a first shell of residue that is instead is blue, so mild stabilizing residues. Then there is a second shell of residues that instead are stability weakness again, and then the third one that is stability strength, and from that shell on they are all in average, they're all stability strength. We also did the same analyzing the evolutionary conservation of the residues. Basically, we ran conserve on all the 550 enzymes, and we plot again the average conservation as a function of the residues distance from the closest catalytic site. And we see also here a kind of oscillation, but here the interpretation is a bit more difficult, because when we look singularly to the enzyme, this oscillation is not so clear. So we are still analyzing the data to understand if this is a general behavior and what is the sense of this general behavior also. And another problem that we tackle, I mean, is a long-standing issue about the cold adaptation of enzyme. We just apply to our approach to have some insight also in this problem. So, I mean, the problem is that the enzyme catalytic rate depend on the temperature. And so if you, for example, you take a mesophilic enzyme that work at 37 degrees and you put it at 5 degrees, there is a drop in the catalytic rate of a factor between 10 and 100. So, I mean, in the literature, people from decades ask how enzyme at low temperature can maintain the catalytic activity, and there are some hypotheses. There are people that say that there is a weakening of the catalytic site region, while more recent investigation by John Akvist and collaborators then instead tend to say that there is the surface that is outside the catalytic region that is instead involved in this maintenance of the activity at low temperature. So we have the enzyme, we have all the data, and so we look at the catalytic site for psychrophilic and thermophilic enzymes. So we basically split, we take a subset of the 550 enzymes that we have in our data set, taking all the cold data and those that have environment in host with the environmental temperature below 25, and those that are above 40 degrees, and then we plot the same graph, so delta G as a function of the distance, and we see that the catalytic side of the cold adapted enzymes are weaker with respect to the catalytic site of the thermostable enzyme. So more in agreement with the first hypothesis that with respect to the second, that in which they claim that there is actually no difference in the catalytic site of enzyme or of psychrophilic enzyme with respect to thermophilic one. So I think I finish with that. So let me just summarize. With our formalism, we are able to study stability patterns, strength, and weakness in proteins, and by applying this to the enzyme, we clearly see some stability oscillatory pattern in the catalytic region, and then applying to the psychrophilic enzyme, we further give some insight about the fact that psychrophilic enzyme have a weaker catalytic side with respect to thermophilic homologous. And now what we are currently doing basically is using what we learn in this investigation to improve our enzyme design algorithm that we have in our lab. And so thank you for the attention.
One quick question. Have you considered comparing more methods, let's say, with all the many available methods, like single point mutations and doing some in silico saturation with genesis to see how they compare?
Not yet to do this analysis. I mean, there are some investigation to slightly similar approach, but I mean, here we don't consider. I mean, there are two different approach. I mean, people from, I don't know if you are familiar with the Walliness group and all the frustration guys, they use mutation frustration. So there are two different things. I mean, there are my approach and I mean doing mutation, because we also have tool to do mutation of the site, but not necessarily give the same result, but it's a good idea, and we have in mind to test that.
Thank you very much.
Thank you.